In this video, we will be discussing types of the signal transduction cascade. And they are G-protein signaling, tyrosine kinase signaling, and cyclic guanosine monophosphate signaling. Let's discuss G-protein signaling. Activation of alpha-1 adrenoceptors, mediated by GQ proteins, and phospholipase C, leads to formation of the second messengers inositol triphosphate, which increases the cytosolic calcium concentration, and diacylglycerol, which activates protein kinase C. GQ, protein-mediated alpha-1 adrenoceptor activity, also activates calcium-dependent potassium channel. The resulting potassium outflow hyperpolarizes and relaxes target smooth muscles.
Protein kinase G mediates smooth muscle relaxation and vasodilation.
Moving forward, activation of alpha-2 adrenoceptors, mediated by GI protein, inhibit adenylate cyclase, so decrease cAMP synthesis and phosphokinase A. At the same time, increase the open probability of voltage-gated potassium channels, leads to hyperpolarization. When coupled with GNOT protein, activated alpha-2 adrenoceptors also inhibit voltage-gated calcium channels and decrease calcium influx. In case of GS protein, adenyl cyclase activity increases, leads to increase in cAMP and also phosphokinase A. Moving towards examples. Example of cAMP pathway are glucagon and epinephrine. Example of phosphatidylinositol for 5-bisphosphate pathway are vasopressin and epinephrine. Moving towards receptor tyrosine kinase. Receptor tyrosine kinase are plasma membrane receptors and have an enzymatic activity. Tyrosine kinase is an enzyme that catalyses the transfer of phosphate group from ATP to amino acid tyrosine on substrate protein. The receptor tyrosine kinases are a large superfamily of receptors that function as the receptors for a wide array of growth factor, including epidermal growth factor and fibroblast growth factor. As we have discussed that receptor tyrosine kinase are plasma membrane receptors. They have the ligand binding site, where ligand binds and initiates the activation of pathway. Now, after binding to ligand, these receptor tyrosine kinase moves close to each other, and dimerization occurs. Now, it is called as dimer, but it is unphosphorylated and inactivated receptor tyrosine kinase. ATP converts to ADP by transferring phosphate group in presence of tyrosine kinase. This is called as phosphorylated dimer and activated receptor tyrosine kinase. Inside the cell, there are inactivated relay proteins. Relay proteins get activated by activated receptor tyrosine kinase. It leads to cellular response. This is how receptor tyrosine kinase gets activated by kinase enzyme. Moving to cyclic guanosine monophosphate signaling. Pathway hormone binds to receptor on cell membrane. There is also a soluble receptor in the cytoplasm. Receptor has intrinsic guanylate cyclase activity. Guanosine triphosphate converts to cyclic guanosine monophosphate. Cyclic guanosine monophosphate activate protein kinase G. So, protein kinase G mediates smooth muscle relaxation and vasodilation. No G protein required. Now let's discuss the examples. So, examples are atrial natriuretic factor receptor on cell membrane. Nitric oxide diffuses across cell membrane and binds soluble receptor. This was all. In our next video, we will talk about the examples of signal transduction cascade.
Explore our extensive library of over 1,800 video lectures to learn about a wide range of topics. Only on sqadia.com.